Okay, so we can start. Our topic is mode of action of pesticides, and as I've said, we cannot actually cover all of these pesticides on the mode of action. So we are going to use some examples so that you can understand what we mean by mode of action of pesticides. The mode of action is actually the biochemical and physiological processes that lead to the destruction of the pest. So if we apply a pesticide targeting the pest, inside the pest we are going to trigger some reactions which are biochemical and physiological processes. This then could lead to the death of this pest. So we are going to look at such reactions, and we are going to focus on organophosphates, carbamates, organochlorines, pyrethroids, and triazides. During this course of discussion, it's unavoidable to use the chemical structures, but don't worry much about them. Just focus on the concept. So we are going to start with organophosphates. These pesticides are widely used to control insect pests. They tended to replace organochlorines because of the persistence problems and the pollution of DDT. DDT is an organochlorine, and these tend to last in the environment for a long time. So people are ready to switch to organophosphates. Frequent sprays are necessary during the course of growing season if you are using organophosphates, because they tend to disappear within the environment. Organophosphates are very effective on aphids. This is the basic structure of the organophosphate. At the center of the molecule we have got phosphorus, and phosphorus is attached to what we can call R1O and R2O. The R1 and the R2 are representing the methyl and ethyl, whereas the O is representing oxygen. Where we have Y, we can either have oxygen or sulfur. Depending whether we are having oxygen or sulfur, it can change the properties of the organophosphate. Then X represents a specific organic group, which can be complex, but as long as we can remember that structure, it's fine. We can have this example. It represents the molecule of the organophosphate. So on the organophosphates, the two other groups are usually methyl and ethyl, like what I said, and are the same in one molecule. What we mean is this. These should be the same. X is a complex aliphatic. It means there are a lot of carbon atoms there, and X is referred to as the living group. It can be POX or PSX, O and S representing oxygen and sulfur respectively. Now we can start with a group known as the orthophosphates. The orthophosphates are represented by that basic structure. You can see on the orthophosphates there is no sulfur. P is surrounded by oxygen molecules, and these oxygen molecules are attached to the other group, which could be ethyl, methyl, and of course we have the X, which represent other structures. So the basic orthophosphates are represented by that structure. We have got examples of insecticides under orthophosphates. We have got chlorvinphos, dichlorphos, mevinphos, and phosphamidone. Another structure for these organophosphates, we have got the thione phosphates or the phosphothionettes. This is the basic structure. If you look at the previous one was surrounded by oxygen atoms, the orthophosphates. Moving on to the thione phosphates, we now have a sulfur. This makes thione phosphates. These are the examples. Maybe you are familiar with them. Bromophos, diazinon, phenylthione, parathione. These are quite toxic. Then primiphos. And you can have methyl or ethyl groups. Then we have got thiophosphates or phosphothiolates. If I move back a bit, you can see where we are having the position of sulfur. Now you can see where we have sulfur, but we have got a double bond on oxygen. That is the difference, slight difference. Dimethyl S-methyl, oxydimetone S-methyl, famidothione. These are the examples. Then phosphonates. Now on the phosphonates again you have oxygen surrounding the phosphorus, but we have the X group directly in contact with the phosphorus. This is what I mean. You can have here, you have got sulfur, but when you have X attached to here, like this one, you have phosphonates, trichlorophone and butyrate. So this is the structure which you should be familiar with. This one is a bit complicated. Pyrophosphoramides. Shradan is the example of the organophosphate. Now we have enzymes which can attack organophosphates. We have got phosphatases, a type of A esterase, microsomal monoxygenase, which is basically cytochrome P450, or you can simply call it cytochrome P450. Then carboxyl esterase, another enzyme which can attack these organophosphates. We have got glutathione S-aryl transferase, also glutathione S-alkyl transferase. All of these enzymes can attack organophosphates. Here we are just showing you some parts, what we are calling the X group, which is joined to the sulfur now. Showing with arrows, we can indicate where these enzymes can act. For example, microsomal monoxygenase and the carboxylase esterase. They can act on that. On this one, microsomal monoxygenase can hit at the hydrogen there, while carboxylamidase can hit at the oxygen there. Now, our interest really is the mode of action of organophosphates. How do they kill the pests, especially the insect? What you should remember is that organophosphates affect the nervous system of an insect. That is their point of action. It is the nervous system. Now, within the nervous system of insects or even animals, we have an enzyme known as the acetylcholine enzyme, ACHE, which resides within the nerve tissues and the neuromuscular junction, where the nerves touches the muscles. That is the neuromuscular junction. Now what happens is that this enzyme, acetylcholine enzyme, can be attracted to the organophosphate. So it can bind to the organophosphate. And it's a sort of a permanent binding structure. So the acetylcholine enzyme will no longer be available. Now the responsibility of acetylcholine enzyme, sorry about that, is to destroy the neurotransmitter acetylcholine. Now, within the nerves of the insect, we have got acetylcholine. Now, its presence may allow the passage of a signal so that probably the insect can jump or move or fly. as a result of these signals, depending on what the insect is responding to. But if we apply the organophosphate, that acetylcholine is now going to be that enzyme is now going to combine with the insecticide. As a result, there will be excessive acetylcholine within the nerves. So the signals will continue to be fired within the insect. If the insect was flying, it's not going to stop flying. It will fly until it dies, because the signal says it should fly, because there's nothing to stop that signal. So the enzyme will not be available to destroy acetylcholine, which holds the signal. This is how the insect can get killed. Here we are just showing you the structure of the nervous system. That is the neuromuscular junction, where you have the nerves attached to the muscle fiber, and the signal can be transmitted via that nerve. But it is at the end of the nerves where it touches the muscle. That is where we find the acetylcholine. We also find the enzyme acetylcholine enzyme, which destroys acetylcholine, so that it breaks the signal or message under normal circumstances. But in the presence of the insecticide, signals keep on coming, so that the insect keeps on repeating some movements, which leads to its death. Again, here we have got the structure of these nerves, which transmit signals. Maybe you met this when you were doing biology some time ago, I don't know. Again, this is where you have nerves which are joined. Where they join is called the synapses. Now, at the synapses area, this is where we find the activity of acetylcholine enzyme and the acetylcholine, which are responsible for transmitting the message or the signal for the insect to do something, either to run or to fly. But if it comes into contact with an organophosphate, it continues to do the action. If it was flying, it will continue to fly. If it was moving, it will continue to move, because of what is happening. Here I have got 3 diagrams, or 3 compartments. You don't have to memorize these compartments, but I am trying to show you where you see those gaps in the first compartment, probably to your left, it represents a normal situation where you have the production of acetylcholine enzyme and acetylcholine production, which transmit the message. After transmitting the message, then the enzyme destroys acetylcholine, which carries the signal, and everything is normal. If you put in an organo phosphate to the insect, if you apply it, what will happen is that the enzyme is going to combine with the organophosphate, and it's not going to, the enzyme is not going to destroy acetylcholine. So acetylcholine will now accumulate in that space, meaning that the signals will continue to be fired. Later on the right, I shall be talking about some other, I think the carbamates, which also act on the acetyl coenzyme A. In this case, they don't necessarily bind, but they compete for acetyl within that sniper's space. I will talk about it later. Well, here it's a chemical representation of what really happens. If you are interested, you can go through those things. Maybe you will get a better picture. Just try to read what is happening there. Here we have simplified the mode of action. You can see where we have put a circle to the right of the slide, where we are saying organophosphates are ACHE inhibitors or acetylcholine enzyme inhibitors. As a result, there is an increase in acetylcholine in the synaptic cleft, the one which is shown at the bottom. Then we have a lot of acetylcholine in that space. Then there is overstimulation. After overstimulation, then there is expression of some symptoms of organophosphate poisoning. So if you read this slide, probably you can easily remember the mode of action of organophosphates. It's meant to simplify what organophosphates do. This one is illustrating the effect or the impact of phosphate poisoning. It could be to humans. I'm not going to spend time trying to describe the symptoms, which are very clear on this slide. You'll be able to remember them. We move on to carbamates. The carbamates are an interesting group of pesticides. they are derived from the carbamic acid. These come from phyllostigmine, which is naturally occurring choline esterase activity. It is actually present in a plant known as the calabar bean, Phyllostigma venosum. This plant is common in West Africa. Actually, it is used traditionally by people in West Africa. If someone dies, they believe that the person has been bewitched because of this witchcraft, which tend to exist in some communities. But how would they deal with such things? They would use that plant, calabar bean, which contains carbamates. So they prepare a concoction which they will give to people, right? If somebody dies after consuming that concoction, they will say that is the person who is actually who is guilty. But if somebody vomits and is not affected, then the person is innocent. What a way of doing things. So you'll find that normally excessive acetylcholine could actually kill victims, because they would be administering, a pesticide to somebody. It's like saying drink a pesticide. If you survive, you are not guilty. If you die, then you are guilty. So this is the brief history of the carbamates. Here we've got examples. The one which is, common is cabril, primicab, carbofuran. I mean, you may have come across these. They originate from the carbamic acid. We have these several: arudicab, oxamil, methomyl, proper care. I mean, some of you are the experts in these pesticides. Some could be nematicides. Maybe you'll correct me on that one. Now let us look at the mode of action of the carbamates, more or less similar to the organophosphates. But yeah, they will combine with the acetyl coenzyme A. Acetylcholine. I don't know why I'm interested in acetyl coenzyme A. Acetylcholine enzyme. But they are going to compete with the acetylcholine. So acetylcholine will compete with the carbamates for the enzyme acetylcholine. Well, we'll call it acetylcholine enzyme, or enzyme ACHE, if you want to call it that way. So what happens is that because of this competition, some of the acetylcholine will be free and it is going to be found in the synapse space. So the excess acetylcholine would cause continuous transmission of messages, and this can lead to problems. And remember, the mode of action is similar to that of carbamates, which can release, but carbamates, carbamates can release acetylcholine faster than organophosphates. So we may say carbamates are less toxic compared to organophosphates, judging from their mode of action. With organophosphate, you have permanent, fixation of acetylcholine. But with the carbamates are actually competing with acetylcholine to get hold of that enzyme. And during that process, some of the, and it can be reversible, some of it may become excessive and damage the insect. Here I've got 3 columns, and we are trying to illustrate a scenario which is the first column where we don't have a pesticide. Things are operating normally, and we are having another case where we have applied B, carbamates have been applied, and they are now combining with the enzyme, causing problems. With the organophosphates, the last column, the combination is almost permanent, and you are going to have excessive acetylcholine. So, yeah, we are just trying to compare where we don't have a pesticide, where we have those 2 pesticides applied. But the basic information is that when you have a pesticide, it interferes with the acetyl coenzyme A, which leads to excessive acetylcholine, which leads to the transmission of a continuous signal, which will then lead to the destruction of the insect. So indeed, carbamates and organophosphates tend to share a more or less similar mode of action. What about organochlorines? These are some of the most widely, they were most widely used in history. And we have things like DDT, dieldrin, aldrin, heptachloriden, linden, and toxaphene, representing the organochlorines. So these are the structures. Some of them we call them body-shaped structure, but anyway, it doesn't. It's not really important. You've got dieldrin there. It's one of the most toxic substances. Then DDT, it's an oral pesticide. Mode of action, they cause neurotic effects. They're going to affect the nervous system. The DDT type insecticides act primarily on the peripheral nervous system. They act on the nerve muscle system. They destabilize neutral activity. Sometimes you can have leakage of sodium ions through the nerve membrane, destabilizing the negative after potential, which is the language which is used to describe the transmission of a signal. Now, if DDT is present in an insect, the insect gets excited. So the manifestation is hyperexcitability of nerves and the muscles. And this is what kills the insect. Probably you may need to explore some of these issues. Now, here we can measure the signals. Let's say you don't have an insecticide present. The signal is represented at the top. But if there's an insecticide, you get those series, of a lot of peaks which are close together. It means there's pesticide activity within the insect. Let us talk about the pyrethroids, which are obtained from a plant known as Chrysanthemum cineariifolium, which is grown in Kenya and Tanzania. The pyrethroids are also synthesized, and they are used to control pests. These are some of the examples of pyrethroids. Let us talk about the mode of action. The nerve excitation occurs as a result of changes in the nerve membrane permeabilities to sodium and potassium ions, and therefore any effect of pyrethroids can be interpreted in terms of such permeabilities. So the presence of a pesticide will affect the membranes of the nervous system. It becomes permeable, and this affects the movement of these sodium and potassium ions. And it can affect the electrical changes within the nerves. The result is that we have a rapid knockdown effects. If you are using pyrethroids, you have a neurotoxic action. The effects could resemble those of DDT. They can cause hyperexcitation and death. Sodium and potassium conductance are altered when the nerve is poisoned. So the potassium efflux stage of recovery after passage of an impulse lasts much longer in pyrethroid poisoned than normal nerve. So the movement of these ions are the center of the cause of probably death and excitation within the insect. Again, we can measure the pulses, the electrical signals. The first line there representing normal peaks, and when the insect is affected, you have the peaks which are close together, meaning that the insect has been affected. So we have got organochlorines and pyrethroids, which may affect, the membranes, which eventually cause movement of sodium and the potassium, which is not normal. It could be calcium and chlorine, and organophosphates and the carbamates. They affect that space there where you have what we call the snipers of the nervous system. It is the excess production of acetylcholine which will give continuous signals that will eventually kill the insect. Here we will talk on the triazines, although we have done it before. We will only use triazines as an example of herbicides in order to talk about the mode of action of herbicides. These are mainly soil-applied herbicides, but can also be used as post-emergence herbicides. Atrazine, which is in this group, is commonly used in maize as a pre-emergence herbicide. Ametrine is mainly used as post-emergence herbicide in sugarcane. These herbicides have a potential to be persistent in soils. Anyway, these are some of the examples of triazines. Again, examples of triazines. We are not going to unpack these molecules. What you need to remember about the mode of action of triazines is that they affect the photosynthesis of the plant. If you affect photosynthesis, it means the plant is not going to grow because of lack of probably carbohydrates, which are basically manufactured by photosynthesis. But where they affect photosynthesis, it is the photosystem 2, if you would still remember it. They are at 2 points where things can start to go wrong. It's between the primary electron acceptor and an electron acceptor known as plastoquinone. The atrazine will block the movement of electrons at that point. As a result, this can lead to a lot of oxygen, production of peroxides, which will actually destroy chloroplast membranes. No wonder why there are these browning effects on the leaves. Also, there is the production of nitrate reductase, which can accumulate nitrites, which also tend to damage the plant. But if you are interested also in the details, but this may be sufficient. You may want to know that the PS2 herbicides inhibit photosynthetic pathway by binding to QB binding site of the D1 protein complex present in the chloroplast thylakoid membrane. It is the binding which disrupts electron transport system ETS from QA to QB, and it can block the fixation of carbon dioxide, ATP generation, and the production of NADPH2. I mean, these are the products of the light reaction which are affected by these herbicides, if you want to use that approach. And of course, if you affect the light reaction, you eventually affect also the dark reaction, because it uses what comes from the light reaction. There's also oxidation, probably because of production of peroxides, which can lead to the death of the plant. So these are some of the details if you are interested in them. But knowing this is, it's quite sufficient, as long as you know that triazine affect photosynthesis. They can take electrons, leading to the death of the plant. So this is photosynthesis. Between primary electron acceptor and the plastoquinone there at the top, that is going to be affected by the triazines, and the whole process is going to collapse. So here's the summary of the mode of action. Of pesticides, we've done that of organophosphates, carbamates, organochlorines, pyrethroids, and atrazine. Actually, I've tried to simplify this very complicated subject. You can imagine we are dealing with so many pesticides, but at least we can have an idea of what they can do to the pest. That is what matters. So this is the end of the first so let me